Good morning, good afternoon, and a good evening to everyone present here today. I would like to welcome you all to our Life Sciences webinar series. My name is Sahil Gulati. I'm the Applications Manager of Life Sciences at Gatan. Before I introduce our speaker for today, I will briefly go through the question submission process. You could type in your questions in the question panel of the GoToWebinar window, which is shown on the right-hand side of the slide. We will try to answer all your questions immediately after the talk, depending on the time remaining. If we do run out of time, your questions will be taken offline, where answers to your questions will be sent via email. With that, I would like to introduce our speaker. I welcome Sudha Chakrapani, who is Joseph T. Warren, MD University Professor in Case Western Reserve University. She's also the Director of Electron Microscopy Core and the Director of Cleveland Center of Membrane and Structural Biology. Sudha received her master's degree in biomedical engineering from Indian Institute of Technology, Mumbai. She then moved to the US for her doctoral work with Tony Auerbach at University of Buffalo on kinetic studies of nicotinic acetylcholine receptors. Then Sudha went on to do her postdoc research with Eduardo Peroso at both University of Virginia and University of Chicago, where she was trained on EPR spectroscopy and X-ray crystallography. In 2010, Sudha started her independent position at Case Western Reserve University, where she rose to the ranks of full professor and is now the director of Cleveland Center of Membrane and Structural Biology. Sudha's lab focuses on structural and functional studies of ligand-gated ion channels. And therefore, our talk today will focus on gating mechanisms and drug modulation of serotonin-3 receptors using cryo electron microscopy. Without further ado, I will leave the electronic podium to Sudha.
Thank you, Sahil, for the very kind introduction. And also thank you and Gatan for this invitation. I'm very excited to participate in this webinar and share with you a little bit about what my group has been working on using cryo-electron microscopy. So broadly speaking, we are interested in understanding the molecular basis for neuronal excitability. Our focus is on ion channels that are responsible for mediating fast signaling at synapses. So I'll talk today about one particular type of systems that we work on in the lab, the serotonin-3 receptors, also known as 5-HT3 receptors. So they belong to a superfamily of receptors that respond to serotonin. Among these receptors, type 3 is unique because this is the only ion channel member, whereas the others going from 1 through 7 are GPCRs. So where are serotonin-3 receptors and what are their function? They are ubiquitously expressed, but their primary function is in the gut-brain circuitry. So about 90% of serotonin produced in our body is in the gut, and these are produced by a specialized type of cells called enterochromaffin cells that lie alongside the epithelial cells in the gut. So these type of cells are sensors of chemical, electrical, and mechanical stimuli in the gut. And in response to these stimuli, they synthesize and secrete serotonin. And so the serotonin molecules then bind to serotonin receptors, which are present on the nerves within the enteric nervous system, which is embedded beneath the gut wall. And they regulate many of the gut functions, such as gut motility, secretion, visceral perception. In addition, serotonin receptors are present on the vagal efferent nerve, also a part of the enteric nervous system, and they signal directly to the chemoreceptor trigger zone and the vomiting center within the brainstem region. Activating these receptors lead to emetic or vomiting and nausea reflexes. There are many dysfunctions that are associated when there is excess serotonin released in the gut in response to either inflammation, tissue damage, or in response to cytotoxic drugs taken in, for example, in scenarios of chemotherapy or during radiation therapy. And hyperactivity of these receptors lead to many of these pathological symptoms that accompany irritable bowel syndrome, visceral pain, and also anxiety, depression, and bipolar disorders. Understandably, these receptors are targeted to treat some of the symptoms associated with these pathophysiologies. Particularly, 5-HT3 receptor antagonists are used to alleviate nausea and vomiting in patients undergoing cancer treatment. And it's also a primary target in pain management associated with irritable bowel syndrome and other gut dysfunctions. So among the several types of 5-HT3 or serotonin 3 receptor modulators are setrons. Setrons are comparative antagonists of these receptors. So currently available setrons belong to first generation and the second generation setrons. So if you look at their structures, what you see is they all share a common pharmacophore. They have a planar aromatic ring. They have a basic amine N that is embedded within ring. And also linking between these two groups is a hydrogen bond acceptor group. Cetrons are, so as I indicated, they are competitive antagonists. So in the presence of serotonin, they inhibit, so these currents, serotonin-induced currents, and their inhibition is reversible. So what do we know about the structural aspects of serotonin 3 receptors? So the receptor belongs to also another superfamily of pentameric ligand-gated ion channel, and the other members of the family include the nicotinic acetylcholine receptors, the one that Sahil mentioned in the introduction that I worked on as a graduate student. So both serotonin 3 receptors and nicotinic acetylcholine receptors are cation selective channels. Whereas the other members, the GABA and the glycine receptors, are the anionic members of the family. So around 2014, 2015 would sort of represent the tipping point in our structural understanding of these receptors. So there were structures of at least one representative members of each of these subfamilies. And a look at how they are built. So even though they only have a small sequence identity conservation among them, they are absolutely conserved in terms of their thread-dimensional structure and overall topology. So each of these receptors, as the name indicates, is a pentamer consisting of 5 subunits. Either they are homomeric or heteromeric assemblies. And each of these consists of an extracellular domain, primarily composed of beta strands, a transmembrane domain with 4 transmembrane helices in each of these subunits, and an intracellular domain. The extracellular domain houses the neurotransmitter binding pocket that occurs at the interface of 2 subunits. The transmembrane helices has the gating machinery and also the machinery for selective ion permeation. And the second transmembrane helix from each of these subunits come together to form an ion permeation path or the pore of the channel. The intracellular domain is the least conserved across the members of the family. It is more structured in case of cation selective channels, whereas it's primarily unstructured in GABA and glycine receptors. So this domain is important for trafficking and also post-translational modification and regulation of ion function and channel functions. So this region has been problematic from the structural biology point of view, given its unstructured nature and high flexibility. And so this region has been previously truncated or partly truncated to allow for high resolution structure determination. So when we started this work, we decided to approach this from understanding the full length of the receptor, given the important role of the intracellular domain in regulating many of important biological properties of this receptor. So at a functional level, the channel can exist in at least three fundamental sort of conformations. A resting state, which is a non-conducting state of the channel. This occurs in the absence of the neurotransmitter. When serotonin binds, it transiently moves to an open conducting conformation. When you're looking at an electrophysiological recording, you see activation current at this point. And the channel in the open state bound to the neurotransmitter is transient. So it eventually moves to the desensitized conformation, and you can see that from the current TK. And the desensitized state is a non-conducting conformation as well. Although both resting and desensitized states are non-conducting, from a pharmacology point of view, they are very different in that they have different affinities for serotonin, they have different affinities for the drugs that modulate these receptors. So an important approach towards targeting these drugs for therapeutic uses is you wanna design, or you want to look at a drug that has that is targeting specific conformations. So it is important that you start off with having at least these initial states or initial conformations of the receptor. So what did we want to understand? We wanted to first understand the conformational changes that underlie serotonin activation and desensitization in a full-length receptor. We wanted to understand what is the structural and functional role of the intracellular domain, and how do modulators affect 5-HT3 receptor function. So at a functional level, what we see is that just like in the cartoon representation that I showed you in the earlier slide, the channels are at rest, non-conducting when there's no serotonin. When exposed to serotonin, they activate and then they desensitize. So a dose response curve indicates that beyond 30 micromolar concentrations or so, you have a saturating response from these receptors. And as shown earlier, serotonin, granisetron is one example, inhibits the response from serotonin in a reversible manner. We started with expressing full-length receptors, and we started with a homopentameric form, and again, simplicity at this point. And we expressed this in SF9 insect cells using baculovirus expression system. Membranes were extracted with C12E9 detergent. We, in this case, used a 1D4 tag for affinity purification of the receptors. So the purified protein here, the peak corresponds to a pentameric channel. We deglycosylated using PNGSF, so to aid in the quality of grid preparation. So this is a collection of some of the structures that we solved of 5-HT3A receptors in various conformations. So we started out with the apo state, which corresponds to a resting conformation. In the apo conditions, we resolve just one conformational state. In the presence of serotonin, however, it was interesting that we isolated two distinct conformational states, which we call state 1 and state 2. And I'll walk you through what these two conformational states likely represent in terms of the scheme that I showed you earlier on. And we were also able to solve several cetron-bound conformations of the receptors at varying resolutions. So I want to highlight here that some of these maps are updated to what we previously published. So all the cetron-bound structures right here and those of serotonin-bound structures were all imaged using our K3 camera here with bioquantum energy filter at Case Western Reserve University. And a 3D movie, I hope it works. Yes, it does. So a map of 5-HT3 receptor bound to one of the cetrons, shown in a yellow gold color right here. So the extracellular domain of the channel. So we were able to resolve individual side chains, allowing us to model the drug receptor interactions. So the transmembrane domain and a part of the intracellular domain. Here, I want to contrast this with a glycine receptor structure that was also solved by the lab to highlight the differences. Although you have quite a bit of conservation in the extracellular domain and the transmembrane region, as expected, we didn't see much of the, or pretty much any of the intracellular domain, given that it's mostly unstructured. 3D model building off from the reconstruction. So what we see here in these green here are glycan density. So serotonin 3 receptors has 3 sets of glycan per subunit. We're able to see resolve a bit of each of these glycans. The transmembrane domain, we resolve a couple of lipid molecules that were bound and came through the extraction process, and the intracellular domain. So we were somewhat disappointed in that even though this is a full-length protein, we did not resolve much beyond what was previously solved in the crystal structure. And this was also sort of expected because this is the region that is unstructured. The top and the bottom view of the receptors. So what I'll show you in the next couple of slides are conformational changes accompanying this receptor in the presence of serotonin, which extends through 165 angstrom and extends across the entire length of the receptor, and allows us to understand what is the basis for this crosstalk that occurs between the extracellular domain and the intracellular domain via the transmembrane region. And here, what we are seeing is a cross-sectional view of only 2 subunits, showing you the ion permeation pathway through the receptor. So this is the apoconformation, and the channel shows constriction within the extracellular domain and 2 notable constriction within the transmembrane domain and an extensive constriction within the intracellular domain. So this is plotting the pore radii along this pathway. And what we see here, the central constriction within the transmembrane domain is a ring of leucine residues that form the activation gate. The dotted line right here is a radius of a hydrated sodium ion. So constriction below the dotted line will represent a barrier to permeating sodium ion. And this right here is a selectivity filter of the channel, which gives it the property of cation selectivity. It is a ring of glutamate residues, which is lying at the interface of the transmembrane domain and the intracellular domain. And the patch of hydrophobic residues right here at the bottom of the intracellular domain also presents another barrier for ion permeation. State 1, which is a serotonin-bound conformation, shows an expansion of the constriction within the extracellular domain. Each of the constrictions within the transmembrane domain is also further widened, as you can see here from the pore radii. There is not much change within the intracellular domain, however. State 2, another serotonin-bound conformations, again widening of the pore in the extracellular domain, further widening of the transmembrane helices, and you see a notable formation of this vestibule at the intersection of the transmembrane and the intracellular domain. And also noteworthy is that the patch of hydrophobic residues stay together within the intracellular domain. Right here is a view of the M2 helices which line the pore, and you see a widening right here in state 1 and further widening in state 2. It is also interesting to note that a serine residue that is present right here causes the bending of the helix in the state 2 conformation. So when you're looking at the pore from the top, you see a rotation and a further rotation in state 2. And since this bending of the M2 helix was stabilized by a serine residue, we decided to mutate to see what is its effect on the stability of the open conformation. So this is wild type currents, the channel activate and desensitize. When mutated to a threonine, we see that this rate of decay of current significantly slows down, suggesting that we are affecting the stability of the open conformation. We then looked at the serotonin binding pocket in state 1 and state 2 conformations. And what we see here is a nice density for serotonin at the interface between two subunits. So this is one of the subunits, this is the adjacent subunit. And this allowed us to model serotonin in both these conformations. A close look at the binding pocket reveals that serotonin is within a cage of aromatic residues. The role of these residues were previously characterized by electrophysiological recordings and many other type of binding studies. So this was no surprise that these residues are involved in an interaction with the serotonin molecule. And we then wanted to understand what's happening within the protein in response to binding serotonin. So here is an overlay, and just shown are two subunits, adjacent subunits from the extracellular domains. APO is shown in red and state 1 and 2 in teal and gold respectively. And so right here is the binding pocket for serotonin. In response to serotonin binding, we see that loop. So this region right here is referred to as loop C. Loop C undergoes an inward movement. And it was previously reported that agonist binding induces this sort of an inward movement within this loop C or the capping loop. And we also see this in these receptors. In addition to the loop C movement, we see that the overall extracellular domain undergoes a twisting movement that leads to repositioning of loops that are at the interface of the two subunits. And again, these studies are consistent with mutagenesis analysis that had been done for several years, which showed that interfacial loops are important for coupling what happens at the neurotransmitter binding pocket to the pore of the channel. A view of what happens at the transmembrane domain level. So this is looking at the channel transmembrane domain from the extracellular end. And what we see here, so this is the pore lining M2 helices. So you can see in comparison to red, you have a rotation to teal and eventually to yellow. And this sort of a movement is leading to the widening of the pore along the ion permeation pathway. In addition to the changes in the ion permeation pathway, we also see that the other transmembrane helices also undergo repositioning. And a consequence of these repositioning is that it changes the volumes of the inter as well as intra-subunit cavities. And it is important because these cavities, both intra-subunit and inter-subunit cavities, are the binding sites for several of allosteric modulators such as alcohols and anesthetics. And these changes in these cavity might represent why some of these molecules or allosteric modulators bind in a state-dependent manner. An interesting observation from comparing these three conformations is that the MX, so this helix right here that forms a belt around at the periphery undergoes a large conformational change going from APO to state 1 and to eventually to state 2. And also the transmembrane helix M4, which is referred to as the lipid sensor in these class of channels, which is at the periphery of the receptor, also undergoes significant conformational change. So here is a movie that is a morph of the APO to state 2 conformation to highlight what's happening at the level of the pore. The channel opens in an iris-like fashion. It leads to this extension of this MX helix away from the pore axis. And you can see this widening of the channel cavities as well. And a closer look of what allows for this large MX movement is that as the channel transition to state 2, we have the helix M4, which occurs as an extended helix with MA, which is a helix within the intracellular domain, is now split into two helices right here at the junction, which is lined by a glycine residue that allows for this break in these two helices. And this movement then causes post M3 loop to move away from the pore axis, pulling MX helix towards it. We questioned what might be the role for such a large conformational change going from state 1 to state 2. And what we find is that this movement, as a consequence, creates a portal at the interface between the transmembrane domain and the intracellular domain, and which is a likely path for ions to exit. And this was previously suggested by Nigel Unwin's work, where he saw these portals in the early cryo electron microscopic images. And we found that in addition to formation of these helices, for these portals, is that they are formed in a state-dependent manner. In the apo conformation, this portal is blocked by the post M3 loop, so you don't see any opening right here. In state 1, you have the formation of this portal, and in state 2, it's a large portal created. Another interesting observation is that this portal is lined by 3 arginine residues that are part of the MA helix. Previous studies have shown that these 3 arginines are responsible for the unusually small conductance of 5-HT3A homopentameric channels. And it was suggested that these arginines might be causing an electrostatic repulsion, lowering the conductance of the channel. And in support of this idea, we see that these arginines are actually lining the portal through which ions exist. And another morphing of the 2 conformational states to show the global conformational changes that accompany serotonin binding. So binds within this pocket lined by 2 subunits, inward movement of loop C. Here is the widening of the transmembrane helices, opening the ion permeation pathway, creation of a vestibules through which ions exit. So having these conformational states gives us an opportunity to evaluate the conductance properties of each of these states. So in collaboration with Mark Sampson's group at Oxford, they carried out these simulations of each of these states. And also, since these are detergent extracted structures, these simulations allow us to evaluate the stability of these conformations in a bilayer environment. And here is looking at the pore profile of each of these states in a membrane embedded environment in 100 nanosecond simulation timeframes. So we see that the deviations from the original Cryo-EM structure is minimal at the level of the pore. And they also evaluated what is the conductance properties of the channel by looking first at the free energy of water occupancy within the pore. And they find that there are 2 major barriers to water occupancy. So if there is a barrier to water occupancy, it also will be a barrier to ion permeation. And they see that this occurs right at the leucine, which forms the activation gate of the channel in the resting conformation, and the hydrophobic patch of residue at the tip of the intracellular domain. In state 1, this barrier starts to decrease, whereas in state 2, the barrier within the transmembrane domain completely disappears, and in line with the idea that state 2 might represent an open conducting conformation. They then applied membrane potential across the bilayer and looked at ion permeation events during these simulations. They find that in the apo conformation, there is a patch, right? So the water molecules are shown in this light green color, and the darker color is of sodium ion. So 0 right here represents the activation gate position, and this is the position at the intracellular domain. In the resting conformation, there is no water in these 2 barriers, and you see no ion permeation. State 1 represents a state which is similar to apo in the sense that there are very minimal water occupancy, but occasionally there is water moving through this position right here, and along with water you also see ions moving through. State 2, on the other hand, is quite different, and this is a completely wet pore at the level of this position, and frequent ion permeation events are observed throughout the simulation. These results suggest that the state 1 might be a low conductance conformation, which could be a pre-open state or a desensitized conformation. And we also evaluated what happens to the pore when it is conducting in state 1 versus when it does not have any water occupancy. We find that the channel can switch between a conducting and a non-conducting state within the same conformation, by simply moving the leucine side chains away from the pore axis. So it's even the slight rotameric reorientation of this position leads to changes in the conductance property of the channel. And the simulation here shows how the ion permeates through the length of the channel and interacts with the activation gate, interacts with the selectivity filter right here, and eventually exits through the lateral portals. It really, and so the tip of the channel within the ICD remains intact and does not, so the preferred exit path for the ion is through these lateral portals. So I want to spend the next few minutes telling you something about what we have done with therapeutic targeting of 5-HT3 receptors. So I started my talk with talking about cetrons, which are competitive antagonists of these receptors. So we were able to get some very nice resolution structures of cetron-bound complexes with 5-HT3A receptors. So the cetron, being a competitive antagonist, binds in the same pocket as that of serotonin molecule. Again, you may recognize the same aromatic residues that are involved in serotonin binding. We then, so this sort of resolution map allowed us to unambiguously model and the orientation of the cetron molecule within the binding pocket. We evaluated the interaction between cetron and the various residues through mutagenesis, showing that mutations in each of these residues impact cetron binding. We wanted to understand the basis for affinity differences and efficacy differences amongst various cetrons. We solved the structures of each of these cetron complex. They all bind in a similar way at the same binding pocket, but they have differences in their individual binding interactions with the aromatic residues. So these are the different cetron molecules. In this project, we collaborated with Martha Filizola's group at Mount Sinai, where they looked at the RMS deviation of each of these binding poses built off Cryo-EM maps. And they find that most of these are stable throughout the simulations, except for serotonin, which is a smaller molecule with smaller footprint, has a lot more mobility within the binding pocket. They find that each of these cetrons primarily interacts with the receptors through apolar interactions. One of the advantages with these simulations is that they can model in water molecules and look for water-mediated interactions, which were missing in our Cryo-EM model. And they identified residues that make key polar interaction with the drug as well as with the serotonin molecule. So identifying these interactions, allowing us to now modify these molecules to improve the drug receptor interactions. Another thing we noted here is that each of the cetron molecule stabilizes loop C to a different extent when compared to the apo conformation and the serotonin-activated state 1 conformation. So as you can see, so this is the apo and this is state 1, and you can see each of these cetron stabilizes loop C to a different extent. And in addition to that, each of the cetron molecules evokes a certain conformational change that appears to be along the same path as the serotonin activation, suggesting that you have a resting state and you have an activated state, and the serotonin seems to be stabilizing intermediate states along the way. Even though these are all partly activated, it appears, from the structural changes, all of the cetron-bound conformation at the level of the pore are non-conducting, so likely stabilizing certain pre-activated states along the activation pathway. So this is another way of thinking about competitive antagonists, is they do not necessarily evoke structural changes that oppose serotonin. It could be similar, except that these might be intermediate non-conducting state that they stabilize along the way. So just want to quickly summarize what I've told you here so far. So 5-HT3A apo state 1 and state 2 structures reveal the intricate molecular motions that associate with serotonin activation in these receptors. And we show that binding of serotonin causes a quaternary change in all the three domains, the extracellular domain, the transmembrane domain, and the intracellular domain. So looking at these conformation changes gives you some understanding of how binding of serotonin in the extracellular domain influences the intracellular domain's interaction with, let's say, binding partners within the cell, or how post-translational modifications within the intracellular domain may impact the affinity of the receptor for the neurotransmitter. We think that the APO and the state 2 structures likely represent resting and conducting conformations respectively. The annotating state 1 is still tricky. It appears to be a low conducting conformation that may be a pre-open state or a desensitized conformation. Cetron binding sites serve as a druggable site for further development of new line of therapeutics, an area that we are continuing to pursue. With that, I would like to acknowledge the people who've done all of this work. So the serotonin receptor project was initiated by Sandeep Basak in the lab, who's now a faculty at NTU Singapore. And all of the electrophysiological recordings for these studies that I showed you was done by Yvonne Gisheru. And these are other members of the lab who are studying other exciting projects, both pentameric channels and associated receptors. Our collaborations with Thermo Fisher Scientific and our MD simulation collaborators, Mark Sampson's group and Martha Filizola's group, our Cryo-EM Access, and our funding agencies for their support. Thank you for listening in, and I'm happy to take any questions you may have.
Thank you, Sudha. It was a beautiful talk with a lot of implications to drug discovery. We have a few questions rolling in. The first question is that, did you encounter any preferred orientation issues in your workflow? Because it's pretty common with membrane proteins. And if you did, how did you address them?
Yes, I think as you mentioned, this is a common scenario with membrane proteins, and we did too. I think that with serotonin receptors, what fixed the problem for us was using fluorinated phospholine detergent as an additive. And this changes the ice properties, and we found that we could get many more views of the receptor with this additive. In other cases, we started to work with nanodisk preparations, and so that also changed the properties, and so we were able to address that using nanodisk, using reconstitution. And more recently, we've also been using graphene oxide grids, which is also helping in some scenarios. So it's a matter of trying many different approaches, and each system seems to have its own solution, or a different problem and a different solution. Yeah.
Thank you, Sudha. We have another question. Do you have any thoughts on how you can improve the resolution of the intracellular domain and why it is not resolved?
Yeah, that is an ongoing problem, and the reason why we are not able to resolve it is that about 60 amino acids that we are missing in the EM density corresponds to an unstructured region. And our approach is to at least getting a bit of insights into those is either complexing with binding partners that may stabilize that region, and or cross-linking assays. Again, in the works, we haven't gotten there yet, but these are some of the strategies that we are trying to explore.
Okay, thank you. And there's another question about the binding of cholesterol with your molecules. Did you find any cholesterol bound when you resolved the structure of serotonin receptors?
Yeah, so we resolved several lipid densities, the identity of which is not is still ambiguous, and there could be some of these that may be cholesterol molecules, and also in glycine receptors. Again, we find several cholesterol molecules. I mean, several lipid densities again. And in those cases, we do add CHS in our purification, so it is possible. But with the resolution that we have, we have not been able to assign or annotate them so far, but we have to have other kind of techniques, mass spec perhaps, to be able to know what comes bound with the protein.
Okay, we have another question. Whenever you are trying to have the ligand bind to serotonin receptors, which could be cetrons or serotonin itself, do you see partial occupancy in the 5 subunits, or all 5 subunits are bound with ligand?
Yeah, that's a great question. So we okay. So with from functional studies, it appears that you don't need to have all the 5 binding sites occupied to activate the receptor. So it suggests that you may, I mean, Hill coefficient is around 2, so it is likely. So you may have 2 molecules binding will activate the receptor. But we have, so from our studies, we don't, I mean, we have a C5 symmetry imposed in our data processing. So we have a density in all the 5. But also because each of these subunits is so, I mean, this is a pentamer, so you have during particle alignment, again, you have the averaging coming about. So we don't at this point have means to know whether there is partial occupancy in these cases. This issue is better resolved with heteromeric receptors. But in the homomeric, we can't tell at this point.
Okay, and this will be the last question, and the rest of the questions will be taken offline, and the answers will be sent to the people asking the question. Now, the last question is that, in your opinion, is a rate limiting step, what is the rate limiting step in your workflow for drug target interaction studies?
Yeah, I think, you know, in the past, I would have said getting the structures of these receptors would have been the major rate limiting step. But given the way Cryo-EM workflows are these days, the initial issues are with sample preparation. So this is somewhat target specific. But once the target preparation, be it its expression, biochemical analysis, Cryo-EM sample preparation optimization is taken care of, so the data collection, I mean, imaging and data processing are perhaps some of the fastest steps to go through. And I think we spend a lot of time post structure determinations. That's where things sort of are time consuming, validation of these binding pockets, molecular dynamic simulations or electrophysiological recordings or other binding studies to validate these models, and compound development, further optimization of the compounds. And all of those are time consuming steps, I would say.
All right, with that, thank you very much, Sudha. Thank you very much, everyone, for attending the talk. Everybody have a great day ahead.
Thank you so much. I enjoyed this opportunity, and thank you, Sahil and Gatan, for for arranging for this webinar.
Thank you.